Hi there. In today's series of videos, we'll be discussing proteomics, the study and analysis of proteins in a cell. In this video, we'll talk about the general benefits and opportunities of studying cellular processes at the protein level. We'll talk about protein preparation methods. We'll talk about the beginning of the proteomic process, digestion of proteins into peptides, and the use of liquid chromatography to separate those peptides so that they can be analyzed by mass spectrometry. The mass spectrometry steps will be covered in the second video, and data analysis will be covered in the third video. This slide is a reminder of the three biopolymers that we've been studying: DNA, RNA, and protein. And in this one, we'll be talking about mass spectrometry, which is a way to study the biopolymer proteins made up of amino acids. It's worth thinking about why study proteomics at all when we have such amazing techniques for studying RNA and DNA. Well, if we think about the central dogma and extend it one step, that DNA makes RNA makes protein, and protein is primarily responsible for cellular phenotype. So if we're trying to understand biology or the way cells are the way they are, the relationship is most close to protein. The relationship between phenotype and RNA is a little removed, and from DNA, most removed of all. Though as we know, ultimately, the relationship between phenotype and DNA will be every bit as clear as the relationship between phenotype and protein when we know much more about biology. Because not only in principle, but in fact, everything derives from the DNA information. Another reason for studying proteomics is that proteins are the primary effector molecules of the cell. They are the molecules that actually do the work, that are the enzymes, that are the structural components, that are responsible for cellular behavior. They're also the primary point of intervention for engineers or physicians. These are the molecules which most drugs target. In fact, nearly all target proteins. And when we make changes in DNA or at the RNA level, we're primarily trying to change the levels of the proteins. So ultimately it all comes down to proteins for the most part. Finally, post-translational modifications like a phosphorylation event can turn proteins on or off, so that kind of a switch is not reflected in the RNA level of that gene, but you can only learn about its presence and its possible effects on activation of the protein by studying at the protein level. And this slide is sort of a repeat of that. When we study RNA with microarrays or sequencing RNA, we're looking at the production of RNA, the degradation of RNA, and alternative splicing. When we study proteomics, we study all of those, but we also look at translational control, how often an RNA is turned into protein, and how the activity of proteins are affected by post-translational modifications. So it's really a much more complete picture of activities going on inside the cell, although its study is slightly more challenging, again, compared to the great methods we have available for the study of RNA. This slide provides a large overview of an entire proteomics type analysis. In step 1, we're going to have a protein sample. We're going to separate it in some manner. Protein gel electrophoresis is a very good method for separating proteins. Then we're going to digest the proteins into peptides. We're going to separate the peptides. We're going to introduce them into the mass spectrometer so that they can be analyzed. Once inside the mass spectrometer, we're going to do two things. We're going to weigh the intact peptide, and we're also going to weigh peptide fragments. Using these two measurements, we'll have a good idea of what the protein is that was the source of that peptide. Third, once we generate this data over here, we have to interpret it and give it a molecular interpretation. We'll see that we do this by a comparison of experimental spectra with theoretical spectra based on protein sequence or genome sequence. We'll do comparisons, and the best matches will give us an idea of the proteins we've observed in our experiment. The fourth stage, which is by far the hardest, is making sense of the proteins that are observed. I refer to this as the list problem in modern biology. We have a lot of high throughput techniques which produce these long lists of proteins or genes or RNA that are affected, but making sense of those biologically is still extremely challenging. Now I'll just compare the steps that I've just mentioned to you in our formulation versus the steps that are listed in the reading which is provided for this lecture. They describe a series of steps which we can correlate with our step 1, step 2, or step 3. Not much discussion at all is given for step 4. In that paper that's posted online, I've grayed out large sections of the paper so that you can focus on just the text that is readily visible. Now when you consider a proteomics experiment, there are going to be several types of samples that you have to think about. One is a single protein band on a gel. When you analyze that with a mass spec, you can typically observe 1 to 5 proteins present. Because mass spec is a pretty sensitive method, and it's extremely hard to take 22,000 proteins in a human cell and do a simple separation step and actually isolate only a single protein. A second type of sample you might want to analyze is a protein complex. These will typically contain between 10 and 50 proteins, and there may be a great deal of heterogeneity because any particular multi-protein complex may exist in different forms in the cell at a specific time. Finally, you might be looking at total cell lysate, and the most repeated estimates of how many proteins are present at any particular time might be 10 to 15,000 in human cells. And current proteomic methods are capable of relatively easily getting to about 5,000 proteins. But getting beyond that is not done very routinely. This slide highlights one of the advantages of proteomics, and it's a relatively detailed depiction of organelles within a cell. One of the advantages of proteomics is you can, say, isolate mitochondria from a cell, and then you can analyze that proteomically, and you can determine the proteins that are involved in mitochondrial structure and function. In contrast, when you think about analyzing cells at the level of RNA, there's no good way to study those subset of RNAs that contribute to mitochondrial function. So for any subcellular analysis, proteomics might be a method of choice. Ultimately, we might wish to understand cells in 3 dimensions in real time. To achieve that understanding, if it's possible, it's going to require methods that analyze cells with some degree of spatial localization, and proteomics is one of those methods. Biochemistry is one of the oldest biological sciences. Any purification protocol that's been developed in the last 100 years now has the ability to be understood in much greater detail by using the sensitive method of mass spectrometry to analyze the results of any previously developed purification protocol. Now when you consider the different samples, the first one was a single band on a gel. Here you have a purification of a subset of proteins. You run it out on a gel, you see a well-localized band. If you cut that out and put it into a mass spectrometer, you should be able to identify that protein with a very high success rate. And indeed, in the last 10 years, nearly every practicing proteomics laboratory has achieved those success levels. Now, for the study of a protein complex, we're going to utilize antibodies that are specific for one protein in that complex. These type of experiments are called immunoprecipitations. Let me write that out for a second. Here we go. It's immuno because the antibody is part of the immune system. Precipitation is because we're going to put this antibody onto a bead, which is macroscopic, can be made out of agarose, and by spitting it or centrifuging it, this bead, which is heavy, is going to go down to the bottom of the tube. It's going to precipitate any protein that the antibody happens to bind and any proteins which are bound to that protein. So in this case, we have an antibody that recognizes this protein that's a black square. This protein is a part of a complex. In this case, only 2 proteins are depicted. There's that red octagon or circle. And it will get pulled down in addition to the black square when you do the immunoprecipitation experiment. This is sometimes called a co-immunoprecipitation experiment because we're pulling down multiple proteins with an antibody targeting a single protein. And immunoprecipitation of a complex is the source of the gel band which we saw earlier. The ARP23 complex is involved in nucleation and regulation of actin filaments. And here they've labeled many of the members of the complex, cutting each band out at a time. But you'll notice, again, some of the difficulty here. There are many bands which could easily be identified using mass spectrometry, which are not part of the complex. That's because whenever you do an immunoprecipitation experiment like this, you're going to be pulling down many, many proteins in addition to the ones you want. So they're using some intensity information here to help decide about which proteins are really in the complex. And they will also determine the complex by doing the immunoprecipitation against many different target proteins. Maybe they would target this protein, and this protein, and those proteins which are common in all of those immunoprecipitations will be considered to be part of the complex. So now that we have the immunoprecipitation, all the proteins sitting at the bottom of the tube, maybe we've separated on the gel, how are we going to analyze them? Well, the most common method for analyzing those proteins is to digest them into peptides. Peptides are of a proper size are easier to analyze than fully intact proteins, although that is a very fast developing field. And one of the ways we do that is using an endoprotease, which is an enzyme that will cut proteins in the middle of their sequences. Much like a restriction enzyme will recognize a sequence of nucleotides and cut DNA in the middle, endoproteases will cut inside a cell after various sequence elements. Now, trypsin is probably the single most utilized endoprotease. It cuts after positively charged amino acids lysine or arginine, and this has a tendency to produce a large number of peptides between 6 and 20 amino acids, which is probably the sweet spot for the sensitivity of the most commonly used mass spectrometer type. So here we have an example of a protein sequence. On the carboxylic side of every lysine or arginine residue, we'll produce a cleavage. So here, in this case, we would have this peptide, which would be suitable for analysis by mass spectrometry. And this is a larger example. Here's the sequence of an entire protein. And then here below, there are many online tools which will do in silico digests of a protein and will give you an idea of all the different peptides that would be produced after such a cleavage. And as you can see, there's a distribution of lengths and a distribution of peptide masses. And as we'll see in the third video, this is an essential part of interpreting mass spectrometry data, that being the in silico digestion of proteins based on known sequence. So now we've taken proteins, we've cut them up into peptides. We're going to analyze the peptides in the mass spectrometer. The way that is most frequently done in mass spectrometry is to load them onto a column. This is often called a reverse phase column, and this has a hydrophobic media, often referred to as C18, which is going to bind peptides based on their hydrophobicity. It's caused reverse phase because these peptides are loaded in aqueous solvent, which will maximize the interaction of the hydrophobic resin with the hydrophobic nature of the peptides. Then we're going to introduce a gradient of solvents. We're going to gradually over time increase the proportion of organic solvents with the water, so that at the end of the gradient it might be 90% organic gradient, 10% aqueous gradient. Now the organic solvent is hydrophobic, and so that you will serially elute peptides. First, you'll elute those that are least hydrophobic by the inclusion of just a small amount of organic gradient. To break the strongest hydrophobic bonds between the most hydrophobic peptides and the hydrophobic resin, will require the most amount of organic solvent. And by increasing the organic gradient over time, we'll release peptides based on their hydrophobicity. And the goal is to get individual peptides into the mass spectrometer at a single time. Protein samples are so complex that you'll never get just single peptides, but this is an extremely efficient method for separating the peptides in narrow peaks with an even distribution. And it doesn't really matter what the complexity of the sample is. We might also do a method where we're going to separate the proteins based on their size, smallest proteins down at the bottom, largest proteins here at the top. Then we'll cut individual bands. Each one of those bands will be a set of 50 to 500 proteins. We'll digest each band from the protein into the individual peptides, and then we'll load each of the the fractions onto the column one at a time, introduce them into the mass spec. And this might be a method, an approach that one might take for trying to maximize the number of proteins identified in a cell at a particular time. Okay, in the next video we'll study how the mass spectrometer interprets peptides as they enter into the instrument and how they are measured. Thanks for listening.